DNA repair after CRISPR cutting is not at all what people thought. This is a report from the University of California. Despite high hopes and high investment in CRISPR-Cas9 gene editing, scientists still have a lot to learn about how it works in humans. In the latest example, University of California, Berkeley scientists found that people's assumptions about how cells repair the genome after the Cas9 enzyme snips DNA are wrong. The discovery gives insight into why CRISPR-Cas9 gene editing works remarkably well in nearly every cell attempted, though not with equal success in all cells. And it could help researchers boost the efficiency with which cells insert new DNA into the genome to replace a harmful mutation with the correct DNA sequence, for example, and generally tweak CRISPR-Cas9 editing to get the desired outcome. Quote, "If you want to treat sickle cell anemia, your chances of success are inextricably tied to the efficiency with which you can replace the mutated sickle cell gene with the correct one," end quote, said UC Berkeley postdoctorate fellow Chris Richardson, first author of a paper describing the findings. Quote, "If you harvest 1 million cells from a patient and you have 10% insertion rate, that is not as good as if you have 30 to 40%. Being able to manipulate those cells to increase the frequency of this process, called homology-directed repair, is exciting. Gene editing is super powerful with a lot of promise, but so far a lot of trial and error. The way it works in human cells has been a black box with a lot of assumptions," end quote, said lead author Jacob Korn, a UC Berkeley adjunct professor of molecular and cell biology. He says, "We're finally starting to get a picture of what's going on." Korn, Richardson, and their colleagues will publish their findings in the August issue of journal Nature Genetics, available online now. Korn was until recently the scientific director of biomedicine in the Innovative Genomics Institute with joint CRISPR research program between UC Berkeley and UC San Francisco. This fall, he will join the faculty of ETH in Zurich, Switzerland. CRISPR relies on DNA repair. CRISPR-Cas9 is revolutionary because of the precision with which it hones in on a specific DNA sequence out of billions in the genome and cleaves the double-stranded DNA molecule. But after that, it's up to the cell to repair the damage. Repair can happen in two ways. Enzymes can stitch the dangled ends back together, which often results in one or more bases, the building blocks of DNA being added or deleted, disrupting the function of the gene. Alternatively, other enzymes can patch the break with a single strand of DNA that matches the DNA sequence upstream and downstream of the cut. A complementary DNA strand is created to complete the double-strand repair. The former, called non-homologous end-joining, appears to be the most common outcome after CRISPR cutting. The latter, homology-directed repair, happens more frequently in some types of cells than others, and requires the presence of a piece of DNA that can be used to patch the break. Researchers often supply a single-stranded piece of DNA and hope that the cell uses it to replace the faulty sequence with a new one. Both processes are a bit mysterious, however, and no one knows why some cells repeatedly patch in DNA while others do so infrequently. Richardson said, "The enthusiasm for using CRISPR-Cas9 for medical or synthetic biology applications is great, but no one really knows what happens after you put it into cells. It goes and creates these breaks, and you count on the cell to fix them. But people don't really understand how that process works," end quote. To find out which DNA repair enzymes are critical to homology-directed repair after CRISPR cutting, Richardson and Korn employed a technique called CRISPR interference, CRISPR-I for short, to knock out, one at a time, more than 2,000 genes known or suspected to be involved in DNA repair, a function critical to healthy cells. Surprisingly, many of the genes that proved to be important, homology-directed repair dropped dramatically when they were silenced, were involved in an important repair system not thought to be involved in CRISPR repair. Fanconi anemia. The pathway involves 21 separate proteins and is called the Fanconi anemia pathway because if any of the genes for these proteins is damaged, people develop Fanconi anemia, a rare but serious hereditary disease in which the bone marrow cannot make enough new red blood cells. It's associated with birth defects and a high risk of cancer, including a 10% chance of developing leukemia in childhood. Few patients live beyond 30 years of age. The pathway has been known and studied for decades, but it was largely understood to repair one specific kind of DNA damage, DNA interstrand cross-links, where a nucleotide on one strand of DNA bonds tightly with a nucleotide on the adjacent strand, interfering with DNA replication and often killing the cell. Researchers in the 1980s had reported a connection between homology-directed repair and the Fanconi anemia pathway, but it had been ignored or misunderstood, Korn noted. Quote, based on our work, we believe that the Fanconi anemia pathway plays a major role in fixing other types of lesions as well, but is best understood as the pathway that repairs double-strand breaks, Richardson said. After Cas9 editing, the Fanconi anemia pathway is required if you want to insert new DNA, end quote. The importance of the Fanconi anemia pathway in repairing CRISPR breaks throws into doubt some planned CRISPR treatments for the disease itself. Without an active Fanconi anemia pathway, cells may not be able to repair their mutated genes with normal genes after Cas9 makes a cut. In fact, the level of activity on the Fanconi anemia pathway may affect how efficiently CRISPR can insert DNA in a specific cell. The researchers concluded that while end joining is the default repair mechanism after a double-strand break, the Fanconi anemia pathway competes with it, and that higher activity results in more homology-directed repair and less end joining. Cancer treatments. While the findings help scientists better understand the DNA repair mechanisms in human cells, they could also help researchers develop anti-cancer therapies that target DNA repair in cancer cells. Because other factors now appear to be involved in the repair of double-strand breaks, this research expands the list of proteins that could be misregulated in order to screw up DNA repair in cancer cells and make them more susceptible to death. Richardson also found that one of the 21 proteins in the pathway, FANCD2, always homes in on the site of the double-strand break created by CRISPR-Cas9, indicating it plays an important role in regulating the insertion of new DNA into the genome at the cut site. FANCD2 could be tweaked to boast the frequency with which a cell inserts DNA via homology-directed repair. Quote, also, since FANCD2 localized to the site of Cas9 breaks, you can use FANCD2 to map where Cas9 is cutting in any cell type, Richardson said. And he added, if you edit a population of cells and you want to know where the on and off target cuts are, you can just map where FANCD2 is found in the genome and you can find the cuts, end quote. And it goes on to say, Korn said, the whole Fanconi anemia pathway affects the balance between end joining and homology-directed repair. It acts like a traffic cop, so a patient's genotype will affect how you do gene editing. So that's bad news, though, if it goes and creates a terrible type of an anemia. What can I say? This is still new medical technology, and hopefully they'll find how to use it properly. This is by Nature Genetics, and it's on by University of California, Berkeley. Journal reference Nature Genetics on physics.org.